So today we're going to be talking about a topic that's a little bit more difficult and special than the first three topics about enzymes and diffusion. Those were basic stuff, but now this is a little bit more complicated. So, Safa, have you done this like the cell membrane before or not?
Not in depth.
Okay, and what about you, Raid? How much in depth have you done it before? Raid, are you there? Okay, you're unmuted. Okay, you're in the chat. Okay, so you have done this chapter already. So let's see, this is going to be an active recall. So let's see how much you understand it. Okay. So we have two important learning outcomes from this. The first ones are generally about the fluid mosaic models. What are the fluid mosaic models? Why are they called the fluid mosaic models? Everything related to their stability, fluidity, permeability, transport, cell signaling. And then what is this cell signaling specifically? How does this come about? What methods of cell signaling do we have? Then we'll move on to the basics that you've already done with a little bit of complication, diffusion added with facilitated diffusion, active transport with endo and exocytosis. We're going to be using, again, an important component of A-level is the practical. So we're going to be talking about the practical aspect here using these different techniques. So that is one interesting thing in this chapter. So let's see. Okay, so the phospholipid, we've already talked about the phospholipid. So just going over it for anybody who does not remember. I told you guys a phospholipid is just initially a lipid with a phosphate. And now what was a lipid? A lipid or a fatty, of a generally fat molecule was made up of this one glycerol molecule. What was a glycerol? A glycerol is a special kind of alcohol that has three OH groups attached to it. Having three OH groups means that it can attach to three other special molecules, because we know bonding always takes place at the OH. The OH is what helps form the water in reactions where water is expelled. So that's usually the O that's involved in bonding. This O over here is the first one, the second one, and the third one. This one molecule is the glycerol. So like I told you, it's a lipid. In lipids, all of the three O's are attached to fatty acid tails. And what are fatty acid tails? They're just acids. We know the chemical formula of acids from chapter 2 is always COOH attached to a hydrocarbon chain. So these are the two hydrocarbon chains. These are the attachments. A phospholipid is special because one of the fatty acid tail that was supposed to be here is now replaced only by a phosphate group, as you can see here. So that's what's special about this phospholipid molecule. If you understand, you'll also understand that in a fat, what it essentially happens is, so we know the hydrogens and carbons are always non-polar, they're always hydrophobic, right? And anything else, for example, the oxygens are usually polar, but there were no oxygens left here. The fat molecules generally were hydrophobic. Hydrophobic means they have a phobia of hydro, which is water. So they hate water. And philic means loving. So hydrophilic, water loving. Now, what ends up happening here is that the two tails, they hate water, but this head we call hydrophilic. Why do we call it hydrophilic? Because it has a phosphate molecule attached. Look at this one here. We have two tails in our phospholipid, and then we have these this head molecule made up of this phosphate and the glycerol because of the oxygens of the glycerol, right? And that is essentially how we learn to orient it. Now, the purpose of learning phospho, and oh, just adding here, when you have a straight chain, that means you have all single bonds. It's called a saturated chain, and it's usually very straight. An unsaturated chain that has a double bond has kinks in it. What do I mean by kinks? Kinks means that it's curved, it's bended. You know, this stage was going straight, but then a double bond came in, and so it had to be kinked. It had to be curved to a direction. So that's how kinks work. Now you can imagine if you have all of straight chains, how wonderfully that can be packed. But if I have a lot of kinks here, if I put a second double bond here and the kink goes here, then that creates space between these tails. And just keep this one phenomena in mind about kinks, because that's going to add up later in their arrangement. Now, why do I talk about the arrangement? This is one slide we talked about from topic one. I told you, let's start from this image right here. I told you that they have hydrophilic heads and hydrophobic tails. Now, if you put some in water, what ends up happening is the hydrophilic end heads love water, so they'll float on top, but the tails hate water, so they'll float on top of water without even touching the water. Now, that's one phenomena when they're freely floating. What if they're truly dispersed in air, water? What if they have no option? They either have to stay in touch with water or fold upon themselves. So this is what ends up happening. They fold upon themselves to form these structures called micelles. And what are micelles? They're just these small blocks or blobs that form where the heads point outwards to protect any water from coming in to the tails where the tails are staying. These can be cylindrical, these can be circular. Very simple stuff. When these micelles, now imagine there is water on the inside and water on the outside, just like a cell. So, what will eventually happen in such a situation? They will line up both from the outside and from the inside. And in the lineup, the tails will always be inside and the heads will always be outside touching the water. And that is simple as that. So the inner layer, this one layer of these tails and the other layer of these tails, they're both hydrophobic. They're not letting any water pass through. But these two layers of the heads, these circles, are hydrophilic. And so this is essentially how a cell is. Imagine this as the cell membrane. This is the extracellular area and the intracellular area, as simple as that. So essentially, cells are just micelles with double layers, which is why we call the cell membrane a lipid or a phospholipid bilayer. It's arranged in two layers, one layer of this and the second layer of this, the same material, two layers to exclude the inside and the outside, to keep away from water. That's how simply it is formed. Again, this is a little bit detailed. Now you can see here we have our one layer and the second layer. For now, let's exclude the blue parts and not focus on them. These are also components of the cell membrane. So just at this point, you need to understand the lipid, how a lipid bilayer forms, why does it form? It forms because of the polarity, the hydrophobic or the non-polar part inside and the hydrophilic or the loving part outside. And then it is semi-permeable. It means it can let some things pass through and something not pass through. For instance, there's a big giant molecule. Will it let it pass through? Not at all. If there's a very small non-polar molecule on the other hand, will it let the non-polar molecule pass through? It will. Now, all of the passing through is simply dependent on what you witness over here. See, the pores between this area are very small, so obviously large molecules can't pass through as easily. Smaller ones can. First point to remember. Second point to remember, the polarity. That was about the size of the molecule. Now the polarity. Is it positive? Is it negative? A positive molecule will definitely be repelled by this internal bilayer and it will not be allowed inside. It cannot move in. The hydrophilic heads are okay with it, but these two layers, got their two layers, they're very strong together, so they won't let it pass through. However, if a negatively charged particles comes in and it sneaks its way through the heads, then all of this is non-polar, and all of this will let it pass through eventually into here. So now we know that size is an important factor. We know that positivity and negativity are considerably important factors. Charged things will stay if they're outside, and if they're inside, which means ions. That is how our body balances ions in the first place. Sodium ions and potassium ions and all the other ions, they can't just freely move across. They have to stay in their place. But sometimes hydrogen ions, and not all the time, sometimes hydrogen ions do move across. Why is that? Because two things add up. It's very small. It's impossibly small. It's just one proton. So it kind of skips through. So that teaches us one thing. It teaches us that if the size is very small, that it is very strongly negatively charged, then it is sorry, but and it is strongly hydrophobic. That is it has no charge and it hates water, then it can easily pass through. If it's larger and heavily charged, then it can never pass through, and that charge can be positive or negative, inconsiderate of whatever the charge is. Now, that's all about semi-permeability. Just like the word says, some pass through because, so, like I told you, very small hydrophobic passed through. Others did not. So semi-permeable, not impermeable. Impermeable means nothing's gonna pass through. So that's not true, as we can see. Now we know the components is a bilayer of phospholipids, as you can visualize here. What are some important stuff within it? Some important stuff is that animal cell membranes, not plant cell membranes, animal and fungal cell membranes only. Please note it down. Always make some notes on your books. This is an important note. Animal and fungal cell membranes will have other molecules in the cell membrane floating with the phospholipid called cholesterol. Fungal cell membranes have something called sterol. It's another kind of cholesterol, but essentially, animal cell membranes have cholesterol. Now, this cholesterol is also floating here, and here you can see these kinks here. Some of the tails are straight and the others are kinked, which simply means this straight one is completely saturated, this one is unsaturated because it has a double bond and is kinked. The third thing in the cell membrane is proteins. Now, these proteins are very important. They allow many functions of a cell membrane to take place. Those functions that we will discuss later as well, but just putting a hint here, they can form channels across the membranes. These proteins can help us form attachments. These proteins might be attached to, for instance, lipids to form lipoproteins or something like that. They can be attached to carbohydrate chains to form glycoproteins. So it's a complex arrangement that we're going to talk about. So the only thing you need to understand here is that in this structural capacity within the phospholipids and the cholesterols, there are proteins arranged that can be arranged in three manners. One manner is covering the entire length of both the lipids. As you can see here, it's going to cover the entire length, fit right between them. Another manner can only be in one layer, either here or here. So this is one of those one layer proteins. The one that's covering the entire length is called an integral protein. It's a very solid block, integral block. Peripheral proteins are on the outside layer. Usually it can be on the inside layer as well. So covering either one layer is called a peripheral protein, and another kind of peripheral protein is something that exists only on the inside or only on the outside, right? It's not spanning between the membrane. It's only either on the inside or on the outside, a little bit stuck here to make sure the connection persists. But this is how it spans. It doesn't span the entire way, it only spans with it right next to the periphery. Now, it's very easy if I tell you that I've told you this area is hydrophobic and this outer and this part is hydrophilic. So if I ask you this part of the protein, is it hydrophobic or hydrophilic? It's very simple because it's right next to the heads. It's definitely hydrophilic, this one and this one. But the inside, this part, it's definitely hydrophobic. Otherwise, the tails would have never let it be here. It would have kicked it out and made their own little bubbles, just like we can see here. So that's about it from this topic, and this is a revision of from topic 1 as well. Let's move on and see another three-dimensional image. This three-dimensional image shows us how the sphere of a cell comes about. Now this can be you can call it a cell, or you can also call it, remember those lysosomes and small spherical structures we were talking about that have enzymes in the center? Even if you want, you can call it those. But the simple structure is always going to be a bilayer arrangement called a cell membrane, or a member, or that cell membrane can be a cell surface membrane or just a cell membrane. When I say cell surface membrane, I mean outside of the cell. If this is outside of the cell and this is the protoplasm, then this makes this lipid bilayer a cell surface membrane. If this is the inside of a lysosome, like we did in chapter 1, then this is just a cell membrane. It's not the cell surface membrane. So just keeping those small concepts in your mind. Excuse me. Okay, so now moving on again to the structure. If you've understood the structure, you'll understand most of it is phospholipid. Anything that resembles a phospholipid without even reading the labels, it's definitely cholesterol that's stuck in between these phospholipids. This shows that this is an animal cell and not a plant cell membrane. Okay, coming again, look at the proteins. Some of them here, some of them here, some of them have holes in them. So proteins, many different forms. This one, he calls it a transport protein. We're going to come to there, but let's first stick to the important structures. So on the outer surface, if the protein has a carbohydrate branching out of it, we call it a for the carb protein, right? A glycoprotein. If that carbohydrate exists on the head of the molecule of our phospholipid, because it's a lipid, we call it a glycolipid. So two important terms, a glycoprotein and a glycolipid. The only difference is, and it's always sticking out from the outer surface, never on the inner surface. A glycoprotein is when the carbohydrate chain is attached to a protein, can be integral, can be a small one over here. Okay, so can be any shape, any size. And if that carb chain is attached to a lipid head, it's called a glycolipid. The smaller ones, again, cholesterol. Proteins, as you can see, peripheral or integral. And then the last thing is that proteins can also exist with gaps in between them. These gaps make it a transport protein because now substances can flow through it. Why do we need these gaps? We need them for the large polar molecules because the polar molecules were continuously being repelled by this internal layer, right? So how will they pass through? They're going to pass through by this special mechanism. Can you appreciate this green part over here? This green part of this transport protein shows us that only this portion of the protein is hydrophobic and attached to the other hydrophobic portion. Now, this portion is so smart, it's cruelly smart. What it does is it confuses the internal portion into thinking, okay, fine, I'm your hydrophobic brother, let me be here. And so it quietly sits in between, because you see these are independent molecules, they're not bonded together. I haven't talked about a single bond like I told you, the bonds in tertiary primary structure of proteins. They're just molecules being pushed together within water because of their polarities. So now what ends up happening is this hydrophilic head is attracted to this one to this, and this hydrophobic head, hydrophobic side is attached to the hydrophobic ends, and the inside, can you see it's cleverly hydrophilic? So now it can allow other hydrophilic molecules to pass through. And the size of this pore, imagine this as a hollow cylinder. Instead of imagining it as two flaps, imagine it as a hollow cylinder. The inside layer of the cylinder is hydrophilic. Now, the size of this pore determines what molecule can pass in. For instance, a large molecule like potassium or sodium, or a smaller molecule like hydrogen. So that's what the size of the pore will determine. And that's what the cell does. We know that the rough endoplasmic reticulum and the Golgi, like in chapter 1, make these molecules. They specialize in making glycoproteins and glycolipids and these special transport proteins that are moved to the cell membrane, right? And so they decide, for instance, one cell thinks, okay, now I want to have a lot of potassium. So what I'm going to do is make a lot of transport protein channels of the size of potassium, for instance 50, and initially I had only two, and put them on the cell membrane. Now what's going to happen? We're going to have like a 48 times increase in potassium entering the cell, and that's how the cell determines what comes into it and how its molecules are affected. So just keep in mind, how do changes in a cell come about? A cell determines what molecules it's going to put into its semi-permeable membrane allowing cells from outside. For instance, a cell decides it has no energy. It puts a molecule that has a opening that can only facilitate glucose. Now that cell's going to have a lot of glucose, right? So it's simple as that. Even if understanding ions is difficult, understanding glucose is very easy. Now at some point, the cell is filled with glucose. He's like, I don't want any more. What he's going to do is order about lysosomes to go and capture this protein and destroy it so that the cell membrane no longer has this extra protein and no more glucose can enter. As simple as that. And even if he needs glucose in the future, he'll just manufacture it by manufacturing a small transport protein. So very easy. Okay, now transport protein channels are of two types, channel or carrier. So we're going to go into details for those, but for now, is the integrity of the cell membrane clear? Do we understand what the cell membrane is? What are the components in it? How it works? How it manages happens in a cell?
Yeah, miss, it's clear.
Okay, perfect. Okay, now again, processes of joining in. Now, I've been talking about how the cell makes stuff. So just imagine, now this is from the outside, but the same thing occurs from here. So there are two processes. We're going to study them in detail as well. I've mentioned them in topic one as well. Endocytosis and exocytosis, which means taking in and taking out, always occur at the cell membrane by the cell membrane pinching in and forming these blebs. These blebs then carry around these substances in vesicles. Okay, similar occurs from the Golgi. As you remember, there were small spheres formed just like this, and the spheres went in. So imagine how the spheres are going to join in. So imagine if this goes opposite, the sphere comes in, the sphere attaches here. These molecular attractions cause these spheres to open up and align and expel the substances outside. The opposite of it can be true. Substances come in, cell membrane pinches in to form the sphere and enter the cell. So very easy. Just this diagram is here just to clear your imagination of how stuff is moving about. Okay, now what is the fluid mosaic model? The model we've been talking about. So it's very simple. It's called a fluid mosaic model, and it's literally what is in its name. It is fluid. That means it is continuously moving. These layers, they're continuously floating among themselves. They're never steady. They haven't stuck together. Like I told you, no bonding. They're continuously free moving. Yes, they will stop where they see an integral protein because this large protein can't float about, but everything else, this cholesterol, it's going to be moving about. So this is why it's called fluid, because it's not static. It is in fact fluid. And then it is mosaic because it is attached together in a mosaic random pattern. Different types of molecules are put together, and those molecules are free floating, are fluid, right? And in both the layers. So if you strip one layer from here, you will see some of the proteins attached here, some here, randomly attached in a mosaic pattern. What are mosaics? Mosaics are just random assortments of art. So the cell membrane is a random assortment in itself. The phospholipids are the fluid, and the mosaics are the proteins attached into it that are not moving, and so they're like icebergs. So what he's comparing it to is, like, imagine the icebergs floating in the ocean, and so a large protein it's gonna be difficult for it to float. A very large iceberg, smaller ones, relatively easier to move a little bit here and there, but phospholipids, like the sea, moving here and there. Now, once you understand that it is fluid, then you also need to understand what fluidity is affected by. And there are essentially just three things. Now, fluidity is affected essentially just by three things: temperature, cholesterol, and whether these tails, like I told you, are saturated or unsaturated. Let's do these two first, and then let's do cholesterol. Okay, so temperature, you know, if it's fluid and if I give it heat, it's going to move around very quickly, right? It won't hold its shape. If it's cold, it won't be able to move around at all. It will lose all its kinetic energy and will become very rigid. So that is as far as temperature goes. For again, for the tails, if they're saturated, they're very, very thin and straight, very neatly packed, not moving about a lot, rigid like the cold. If they have kinks in them, not as neat. They can move around more easily, they hit each other more easily, move here and there. And so, again, not a very difficult concept, right? Now, cholesterol is supposed to be the balancer. It's supposed to be that thing in the equation that prevents excessive fluidity, whether it's too hot or too cold. It acts basically as a buffer. It prevents the cell membrane from becoming too disoriented. So that's essentially why cholesterol is very important in cell surface membranes, right? And if you remember, where were fats? So we know everything's being produced inside the cell. So just a brain quiz over here. Where are these sterols and cholesterols made within the cell? We've talked about the rough endoplasmic reticulum for the phospholipids and the glycolipids and all the complicated stuff. Where is the cholesterol made? In the smooth endoplasmic reticulum. We've talked about how fats are produced over there. So I hope you have that concept clear as well. So membrane fluidity affected by these three things in which cholesterol is just a buffer. It's not going to let the fluidity increase on high temperatures or decrease on cold temperatures. Okay. Now, let's see the functions. We've talked about these molecules. Let's quickly go over this slide and see what the important structures over here are. We've talked about glycoproteins. Look at this glyco chain attached to a protein. Look at this chain attached. Look at this glycolipid. Why is it a glycolipid? Because it's attached to a lipid, a cholesterol molecule. Carbs are attached to it. So look at this one attached to the lipid molecule, glycolipid again, protein, an integral protein, a peripheral protein, another protein. This is called a surface protein or a type of a peripheral protein. Look at this protein channel, a transport protein. And also look at this kind of a protein extending outwards and inwards. So very special kind of a protein. You don't need to know the details. This is a single layer. This is the second layer. Again, not very difficult, I hope. If there is any question, you guys can ask me. So now we need to understand. We've understood how the fluid mosaic model works, how the permeability is controlled, how these are affected, the phospholipids are affected by different changes. Now we need to understand the different molecules, like these glycoproteins and lipids and normal proteins. They have six functions. The first function is they act as receptor molecules. Now, how are they going to act as receptor molecules? For instance, this special cell membrane has this one single sticking out over here and this second one sticking out over here. And these two are, let's say, it's handles or identifiers. These two, if this cell goes and meets another cell, it's a red blood cell, it goes and touches an intestinal cell to deliver oxygen, that intestinal cell will recognize this molecule sticking out and this molecule sticking out, this glycolipid and this glycoprotein sticking out, and it will say, okay, this one is a RBC. I will let it give me some oxygen just to make sure, for instance, if a bacteria comes in, its receptor molecules are different, so that the body will recognize immediately that these receptor molecules are not the same one as my cells. So now what I'm going to do is activate an immune response. And that's essentially what receptor molecules do. And what are those receptor molecules? Those are these specific lipids and specific glycoproteins sticking out that help you decide that this person is the specific one. Imagine a person. How do you recognize that they are this specific person? By looking at their eyes, face, and nose. So these glycoproteins and lipids and other proteins are the nose, eyes, lips of a cell surface membrane, which help us recognize. So they're essentially receptor molecules. They help in cell recognition, they help in reception, attachment of cell. If I think this is you, I'm going to shake your hand, I'm going to accept this limb that you're extending, this glycolipid that you're extending, right? These can also be enzymes. For instance, these enzymes can break down, for instance, any incoming molecule, any pathological molecule. So they can, because they're proteins, they can also be enzymes. This integral protein, maybe it's an enzyme. It's gonna, if glucose comes in, it's gonna break it down and release the products inside. These can be attachments for cytoskeleton. All of those microfilaments and microtubules might be attached into this integral protein and then inside the cell, forming a network, forming a spanning network. And they can be used for cell signaling. One cell can go and hit the other cell with, for instance, this protein molecule, and that cell is going to recognize, okay, it's dangerous here, let's go on. Let's not stay here. There's a pathology, there's a virus over here. Or maybe if this is let's say it's a white blood cell, it's going to come in, it's going to recognize a virus, and then it's going to go on and call in other cells by cell signaling, call the more white blood cells and tell them to eliminate the virus. So I hope that these functions are clear. What I want you to do, I did not write a lot of text about them because I really want you to go back and read up on them. These functions are very easy, but they're very theoretical. Once you go and read, because there's not much detail in your A-level syllabus, if you go back and read them up, you're going to perfectly understand them. So that's your one task for tonight. Please go back and read these 6 important steps. Screenshot this slide if you have to. Read how they come about, what molecules make them come about. It's very simple. I'm just going to talk about one important one here, which is cell signaling, which is in much more detail than the other ones over here. For the other ones, you can just read them up and you'll definitely understand them. Now, cell signaling can be in many different pathways. Okay, so cell signaling can take place in different pathways, right? Cell signaling can take place now, just following a few of the things here. It can take place when, for instance, a molecule that I'm calling a ligand here. Ligands are stuff that fits into to act into a surface to activate it. For instance, I have a signal which is a ligand. That ligand can act on a receptor on the outside of the cell to lead a cascade of changes within the cell. That if that ligand is hydrophobic, if it's hydrophilic, it can't enter the cell, so it's going to act on a receptor and bring about changes, bring about signal molecules. These signal molecules will lead to protein production, and then those proteins can be secreted outside of the cell or used inside of the cell or given to other cells. If it's a hydrophobic molecule, it can directly enter inside the cell and bring about those changes itself. Now, what is this signal? For instance, it's a hormone. I say it's a specific hormone. It's insulin. Insulin is going to come about attached to its ligand and cause opening and cause making of protein channels for glucose. And now a lot of glucose channels are going to be made inside the cell and attached to the cell membrane. And that's how basically insulin manages glucose levels. When those carrier proteins are attached in the cell membrane, more glucose can enter inside the cell and be used by the cell. And as soon as the insulin is detached from this ligand, more the proteins are stopped being made. And so no more glucose, no more proteins made. The previous ones are being broken down, and so no more glucose can enter. But if it's hydrophobic, like for instance, if it's testosterone or if it's estrogen, it's gonna go directly into the cell and act on the nucleus to increase production of certain mRNA and certain protein. It does not need a ligand anymore. So cell signaling can be in many possible ways. The only thing you need to understand is that a stimulus or a ligand comes in, acts, whether acts on a receptor or does not act on a receptor, acts directly on the target effector cell, can occur in both ways. Now, this transmission of signal from the ligand to the effector is called transduction. You need to understand the term transduction. Transduction in layman term just means that your signal is being converted from one form to another form. In here, it was in a form that the cell could not use to perform a function. So what happens is it comes inside, it makes changes in the cell. These changes make it into a transducible form in which the cell can finally make proteins. Now, this can act on a receptor, on a ligand receptor, or it can be directly by entering directly. So transduction is this signal being transmitted to into the language of the cell. Let's say this was not the language of the cell, and then we used a mediator to convert it into the language of the cell. And this conversion from its own language to the language of the cell is called transduction. So as simple as that. Now, How does this whole system come about? So, for instance, one cell releases ligands. These ligands, I tell you, are large they're very large hydrophilic molecules. Now, they can't diffuse right across the cell wall. So what happens is there are special receptors. They'll attach to these receptors. These receptors will send signal to certain proteins. These proteins will make further proteins which will bring about the response. For instance, they'll make a specific hormone, a specific enzyme, and bring about the response. If another cell doesn't, and why is this ligand channel or receptor response important? Because in cells that so for ligands that are hydrophobic, they will act on every single cell in the body, but hydrophilic cells can be controlled. Ligands can be controlled. Why? Because cells that do not have receptors for them will not be influenced. So for instance, if we don't want this cell to react to testosterone, if in certain bodies we don't want testosterone to act upon certain cells, if testosterone used, it does not use it, but if it did use a receptor, we'll be free from these side effects. But that's why testosterone is dangerous for like female bodies, or it's excessively dangerous for females. Why? Because it's going to enter every single cell. There is no control. On the other hand, insulin does not work like that. Insulin has receptors, so cells that don't have receptors will not be flooded by the response. Only cells that have receptors will be flooded. Again, this shows the signaling molecule from your book. So we have a ligand attaching to a protein. Now, this specific ligand pathway is called the G protein pathway. Now, you need to be able to at least recognize and write something about it. So ligand comes in, attaches to the protein receptor. We've talked about that. Now, why is it G protein? It's this protein receptor is attached to a G protein. As soon as the ligand comes in, the G protein is activated. And when this G protein is activated, it acts on a nearby second messenger. Now you guys, the confusion about how proteins can be enzymes, this second messenger is an enzyme which will lead to break down certain or speeden up certain reactions, leading to certain proteins and other enzymes being formed. Now a cascade occurs. What is a cascade? It's like a rolling ball in snow. One molecule will lead to another, will lead to another, will lead to another, right? As soon as this enzyme is activated, it's going to make more molecules and more molecules and more molecules. And eventually, so many molecules are made that a huge response takes place very quickly. As compared to if it was a hydrophobic molecule, just one molecule would diffuse in and come here and lead to the secretion and transcription and movement. Over here, a cascade of response occurs thanks to the ligand.